Hello everyone, this is Dr. Divya and today's topic for discussion is Western blotting techniques. Western blotting, used to detect specific protein in a sample of tissue homogenate or extract using labeled antibodies. The method originated in the laboratory of Harry Taubin at the Friedrich Miescher Institute, Switzerland in the year 1979. The name Western blot was given to the technique by W. Neil Burnett and is a play on the name Southern blot. The membrane used for this Western blotting is nitrocellulose and supported nitrocellulose and polyvinylidene difluoride. It's also known as PVDF membrane. In Western blotting technique, the method used here is protein of the sample was separated by gel electrophoresis. Separation of proteins may be by isoelectric point, molecular weight, electric charge, or a combination of these factors. Most common type of electrophoresis employs polyacrylamide gel, PAGE, and buffers loaded with sodium dodecyl sulfate, SDS. Proteins are transferred from the gel onto a membrane made of nitrocellulose or polyvinylidene difluoride (PVDF) for accessibility to antibodies. Transferring by electroblotting uses an electric current to pull protein from the gel into the PVDF or nitrocellulose membrane. The proteins move from within the gel onto the membrane while maintaining the organization they had within the gel. The uniformly and overall effectiveness of transfer of protein from the gel to the membrane can be checked by staining the membrane with Comois brilliant blue. The membrane is probed for the protein of interest with a modified antibody which is linked to a reporter enzyme. When exposed to an appropriate substrate, this enzyme drives a colorimetric reaction and produces a color. It is detected in two ways. One is direct detection and indirect detection. In direct detection, uses a label primary antibodies to identify the target protein, whereas in indirect detection, uses an unlabeled primary followed by labeled secondary antibodies. Western blotting application. It is used for the confirmatory HIV test, employs a Western blot to detect anti-HIV antibody in a human serum sample known as ELISA. This technique is used for the detection and analysis of proteins. It is also used for diagnostic of various diseases like viral and autoimmune disease included. And these are all the book for the references. Thank you.